How do gene fusions drive Ewing sarcoma tumor development? Imagine a tiny mistake in the way our DNA is arranged that can lead to a cancer growing in the bone. In Ewing sarcoma, this mistake involves a specific gene fusion that turns normal genes into powerful drivers of tumor growth. This gene fusion happens when parts of two chromosomes break and reattach in a way that creates a new, abnormal gene. The most common fusion involves the gene called EWSR1 on chromosome 20 and a gene from the ETS family called FLI1 on chromosome 11. When these two fuse together, they form a new gene called EWSR1 FLI1. This fusion gene produces a fusion protein that acts as an abnormal transcription factor, meaning it can turn genes on or off in ways that are not normal. This fusion protein is central to how Ewing sarcoma develops. It binds to specific DNA sequences called GIGA microsatellite repeats, which are normally inactive or have little role in gene regulation. The fusion protein turns these repeats into active enhancer regions, which are like switches that turn on genes. These genes promote cell growth, survival, and division, fueling the tumor. At the same time, the fusion protein can suppress genes that would normally prevent tumor growth, creating a perfect environment for cancer to thrive. The formation of this fusion gene is not always simple. It often results from a complex process called chromoplexy, where multiple DNA rearrangements happen suddenly, disrupting several genes at once. This process tends to occur in parts of the genome that are actively being copied and used by the cell, making the fusion more likely to happen in aggressive tumors. The fusion gene is always found at the heart of these rearrangements, highlighting its key role in starting the cancer. The fusion protein does more than just turn genes on and off. It influences how DNA is packaged in the cell, affecting chromatin structure, and it can alter how RNA is processed and how proteins are made. These broad effects help the tumor cells grow faster, avoid normal controls, and become more resistant to treatment. While some additional mutations in genes like TP53 or CDKN2A can occur, they are less common and usually make the disease more aggressive. Most of the tumor's behavior is driven by changes in gene regulation caused by the fusion protein rather than multiple genetic mutations. Understanding how this fusion gene works is vital for developing targeted treatments. Researchers are exploring ways to block the fusion protein from binding to DNA or prevent it from activating cancer-promoting genes. Although current treatments for Ewing sarcoma include chemotherapy, surgery, and radiation, future therapies might focus on disrupting this fusion protein directly. This knowledge helps us see how a single gene fusion can have such a profound impact on tumor development and offers hope for more precise, effective treatments in the future. Always remember, if you or someone you know is affected by Ewing sarcoma, consulting healthcare professionals is essential for personalized care and the latest treatment options.